Good morning to all. Welcome to the Diamond Group of Institutions, NEET and K-CET online classes. Today's class, photosynthesis in higher plants. So in that, some previous important questions, concepts we have to discuss. So in that some the law of limiting factor for photosynthesis was announced by. So here, the law of limiting factors. So this concept we are discussed at the factors affecting of photosynthesis. So here, law of limiting factor. What's the meaning of the law of limiting factor? So first you want to know law of limiting factor says as if a chemical reaction more than one factor is involved in a chemical reaction, more than one factor is involved, the rate of the reaction determined by one factor, it's near into the minimum value. Which of the factor can determine the rate of reaction that is near into the minimum value, that is called as a law of limiting factor. Means so in a group or in a chemical reaction, a group of the factors, more than one factor is involved. Example you are taken as a photosynthesis. Photosynthesis can depend upon the several group of the factors like as a light, CO2, temperature, water, those are. So in this, more than one factor is involved. So the rate of the photosynthesis can determined by the one factor that is near into the minimum value, that says as a law of limiting factor. That law of limiting factor was given by Blackman scientist. And remind option D, Robert Hill, he was proposed the Hill's reaction. It says as splitting of the water in the presence of light and then form as a protons, electrons and oxygen molecules will be released. That is explained by Hills. Rubin and Tromen, these are two scientists are experimentally proved by the using of radioactive oxygen molecules, releasing of the oxygen from water, not from the carbon dioxide. That is experimentally proven by the Rubin, Tromen and Hussey. These are three scientists. So question was asked as a law of limiting factor. That law of limiting factor was proposed by Blackman scientist. And the next question, Emerson enhancement effect and red drop effect has been instrumental in the discovery of Emerson enhancement effect and red drop effect. So here, before the light reactions, in the chloroplast, thylakoids are present. In the thylakoid group of the photosynthetic pigments are there present. The group of the photosynthetic pigments are we call as a quantasomes. So here in a chloroplast, that is in a thylakoid, single quantasome it was present. So these are the single quantasome it was present. It can update more than 680 nanometers of the light. If the single quantasome, otherwise we are also called as a group of chlorophyll pigments, are also we call as a photosystem. If it's contained as a single photosystem, can update in these are the photosystem more than 680 nanometers of the light it was fall. So more than 680 nanometers of the light we call as a red light. If the red light it was fall on a photosystem continuously, the rate of the reaction will be done to stop. The rate of the photosynthesis will be done to decrease. So decrease the rate of photosynthesis due to the continuously fall of the red light. So that is called as a red drop effect. This is we call as a red drop effect. So means of the red drop effect it is if the thylakoids contain as a single quantasome, otherwise photosystem, that quantasome or photosystem can receive more than 680 nanometers of light. So if more than 680 nanometers of the light is continuously fall on the photosystem, the rate of the photosynthesis, otherwise yield of the photosynthesis will be done to decreased. That is due to the red light. So this is called as a red drop effect. So for the increase the rate of photosynthesis, increase the yield of the photosynthesis, Emerson, one of the scientists, he was proposed the two photosystem concept. The two photosystem concept says as, so one of the photosystem can receive the 680 nanometers of the light, and other one of the photosystem can receive the more than 680 nanometers of the light. So now by the presence of photosystem 2 and the photosystem 1. So these are the two photosystems are present in a thylakoid of the chloroplast. These photosystem can receive the light in a different wavelength. Different wavelength. So at a time light will be going to fall in two photosystems at a different wavelength. The rate of the photosynthesis enormously increased. Increase the rate of photosynthesis because of the presence of two photosystems, that is called as a Emerson enhancement effect. See, it was explained by Emerson scientists, so that's why we call as a Emerson enhancement effect. So Emerson enhancement effect and red drop has been instrumental in the discovery of. So why these are Emerson enhancement effect and red drop effect is proposed. So option A, photophosphorylation and non-cyclic electron transport system. Option B, two photosystems operating simultaneously. So here the two photosystems are there operated simultaneously. One it is 680 nanometers and other one it is more than 680 nanometers as a 700 nanometers. And option C, photophosphorylation and cyclic electron transport system, oxidative phosphorylation. So now it was for the Emerson enhancement effect and red drop effect, it is related into the two photosystem operating simultaneously. It's a two photosystem concept. Then what about the given options? Photophosphorylation and non-cyclic electron transport system. You know that synthesis of ATP takes place in the presence of light. It's called as a photophosphorylation, and the non-cyclic means, so light reactions are occurred by two types, cyclic and non-cyclic electron transport system. So in the non-cyclic electron transport system, water electrons are formed or released by the photolysis of water. These electrons will never come back into the lumen again. So that's why it is called as a non-cyclic. It's not related into the Emerson enhancement effect. And photophosphorylation and cyclic. So in the cyclic electron transport system, water the electrons are released from the photosystem 1, these electrons are they can come back into the photosystem 1 in a cyclic manner. So that's what it's called as cyclic electron transport system. And oxidative phosphorylation. This concept is related or we are seen in a respiration. Here also synthesis of ATP take place, but oxygen will be not involved. So answer for this Emerson and the red drop effect it is D, two photosystems. And next question. Angelman's experiment with spirogyra demonstrated that T.W. Angelman, one of the scientists, he was done the experiment by the taking of the aerobic bacteria, and he was taking green algae, the mixture of aerobic bacteria, that is he was taken cladophora. So he was taken into the group of the green algae and the aerobic bacteria in a petri dish. The petri dish he was kept in a light fall. So here in the T.W. Angelman experiment, a monochromatic light will be going to fall in a prism. The light will be going to split and they can form as a 7 colors, V, G, R. So wherever the light will be going to fall in that region, the group of these algae and the aerobic bacteria was kept out of the Vibgea tellus. Out of the Vibgea tellus, so green algae can receive the it can receive the red light and they can prepare the food materials, and the aerobic bacteria, that is cladophora, can receive the blue light and they can prepare the food materials. Means photosynthesis is take place in a red light and the blue light mostly. So that is explained experimentally by T.W. Angelman. So T.W. Angelman's experiment with spirogyra demonstrated that the full spectrum of sunlight is needed for the photosynthesis. Only red wavelength are effective in causing of the photosynthesis. See now here, The full spectrum of light is needed for the photosynthesis. What are the seven colors are formed? These are the seven colors are required for the photosynthesis. It's not a true. And only red wavelengths are effective in processing of photosynthesis. So red light is required for the photosynthesis for higher group of the plants. But he was taken into the green algae and also the aerobic bacteria. So that aerobic bacteria can receive the low intensity of the light. And option C, only blue light wavelengths are effective. So only the lower group of the organism light as the bacteria can receive the blue light. Both blue and red wavelength are effective. So in a process of photosynthesis, he was taken in a mixture of green algae and the aerobic bacteria suspension. In that, aerobic bacteria can receive the blue light and the green algae can receive the red light. So based on this thing, so both of the blue light and red light is required for the photosynthesis. And T.W. Andelman, he was explained by absorption spectrum and the action spectrums also. So this T.W. Andelman, he says as both of the red and the blue lights are required for photosynthesis. So answer D. And the next question. Mole's experiment shows as moles, that is we are discussed as a introduction of photosynthesis. Some of the experiments we are going to discuss. Those are the experiments. Some of the factors are needed for photosynthesis or components required for the photosynthesis. At the time we are going to discuss the moles half-life experiment. Moles, one of the scientists, he was done the experiment and he was taken a green leaf. That green leaf he was kept in a bottle contain of KOH solution. A plant leaf he was inserted a bottle contain of the KOH solution. Half part of the leaf it was present inside of the bottle and half part of the leaf is exposed into the environment. So what it says about this? He was taken in a bottle KOH solution, potassium hydroxide. So now these are the KOH solution function. It can update wherever the CO2 molecules are there present. The CO2 molecules are completely absorbed by the KOH solution. So half part of the leaf is kept in a bottle contain of KOH and remaining half part of the leaf was exposed into the environment. That environment is contained as a light CO2. So for the components of photosynthesis, for the photosynthesis, light, CO2 and water and chlorophyll pigments, these are the major components for photosynthesis. Out of that, CO2 also one of the major factor for the photosynthesis, one of the major component for the photosynthesis, that is experimentally proven by moles. So moles half-leaf experiment, what it says? Half part of the leaf was kept inside of the bottle. So remaining half part of the leaf is exposed into the environment. It's contained as a CO2. So now here what is the CO2 is available in a bottle. It is completely absorbed by KOH solution. Here the CO2 is absent. And half part of the leaf, remaining half part of the leaf which is present outside of the bottle, they can update carbon dioxide from the environment. And here the photosynthesis will be done to perform. Preparation of the food materials that takes place. After some period of time, so the leaf will be done to treated with iodine solution. So half part of the leaf, this part is exposed into the photosynthesis. It will become as a blue in color. And this part does not perform the photosynthesis, does not change any color, no color change we are going to observe. That is says as a photosynthesis, means that is the experimental proof. Here, why this part only can change the color? Because here, this part, which are the part it was present outside of the bottle, the leaf can perform the photosynthesis in the presence of CO2, light, and the water chlorophyll pigments. But which of the leaf it was present inside of the bottle, it's contained as all components except the CO2. So the CO2 availability is absent, so photosynthesis is not take place. Food materials are not prepared. If you have done the test with iodine reagent, here does not change any color. So that's the experimental proof by moles of leaf experiment, and CO2 also one of the major component for the photosynthesis. Based upon this experiment, we'll get. So moles of leaf experiment shows as unequal transpiration from two surface of leaf, relation between transpiration and absorption. CO2 is required for the photosynthesis. Mole said as for the photosynthesis, CO2 is required. Without CO2 molecule, they cannot perform the photosynthesis. And chlorophyll essential for photosynthesis. Here, moles of leaf experiment says as CO2 one of the major component for the photosynthesis. Answer C. Yes. Next question. Which of the following represents the correct molecular formula of chlorophyll B? So we have to identify the molecular formula of chlorophyll B. So chlorophyll pigments they contain as a single magnesium atom. These are the single magnesium atoms surrounded by the 4 nitrogen atoms, and these are the 4 nitrogen atoms are they contain as a chiral rings. So this is the head of chlorophyll. So now we have to identify the structure of chlorophyll B pigment. So it's light as a chlorophyll A only. Some major differences between the chlorophyll A and the chlorophyll B pigments. So in a second carbon atom of the second chiral ring of the third carbon atom of the chlorophyll B pigment, instead of this aldehyde, methyl group will be drawn to presence. So that is a major difference between chlorophyll A and chlorophyll B. And when we see the molecular weight of or molecular structure of the chlorophyll A, it is chlorophyll A is C55 H72 O5 and 4 Mg, and the chlorophyll B it is C55 H70 O6 and 4 Mg. So according to the option, as this is a chlorophyll B and this is a chlorophyll A. So chlorophyll B molecular formula they are asked. So here they are given as C55 H70 O6 and 4 Mg. Answer is C. The next question, make a suitable pair. So they are given as again Emerson effect, Hill's reaction, Kelvin cycle, Hatcham-Slack cycle. Emerson effect already we know, we are discussed. It's a two photosystem concept. So for the A it's a D. And Hill's reaction, Robert Hill, one of the scientists, he was done the experiment and he was experimental proven photolysis of water, splitting of the water in the presence of light, and they can form into protons, electrons and oxygen molecule. And Kelvin cycle is also called as a C3 cycle. Hatcham-Slack cycle we are also represented as a C4 cycle. For the correct match for these A, D, B, B and C, C, D, A. So answer D. In the next question, match the site in column 1 with the process in column 2 and choose the correct combination from the options. So column 1 and column 2 they are given as a grana of chloroplast, stroma of chloroplast, cytoplasm and mitochondria, Krebs cycle, light reaction, dark reactions and glycolysis. For this concept you want to know the structure of chloroplast. So here let's see the structure of the chloroplast. Chloroplast it was present in a higher group of the plants, mostly in the form of a circular or oval shaped structure. It's a double membrane structure, cell organelle. It's contained as a inside of the chloroplast. A many number disc shaped structures are there arranged one to one another. So those are we call as group of the disc shaped structures are called as a grana. In this single disc shaped structure, this is we call as a thylakoid. So here the thylakoids are there involved in a light reactions. So this group of the thylakoids are we call as a grana, and in these of the thylakoids are the grana it was present in a liquid-like structure. This is we call as a stroma. So in that grana, light reactions are take place. In the stroma, dark reactions are take place. This is stroma, or in the region of the stroma, biosynthetic phase or biosynthesis of the food materials will be don't occur. So here, what happens in a thylakoids? Thylakoids are they contain as a photosystems, that is photosystem 2 and the photosystem 1. They can trap the light. That light energy is converted into the chemical energy in the form of ATP and the NADPH2. And this is ATP and NADPH2 we call as reducing energy. And this reducing energy can supply, they can transfer from the thylakoids into, that is the grana, into the stroma region. There the dart reactions are take place. Preparation of the food materials are occurring. So now let's see grana of the chloroplast, stroma of the chloroplast, cytoplasm and mitochondrial matrix. So options are, so grana can perform into the light reaction, grana of chloroplast. And the stroma of the chloroplast, they can perform the dart reactions, means it's a biosynthetic phase. It's in this phase, C3 and C4 cytosine, preparation of the glucose molecules are take place. And coming to the cytoplasm and mitochondria. So the cytoplasm and mitochondria options are given as a Krebs cycle and a glycolysis. These two are the structures are we are seen in a respiration in a plants. So now here the mitochondrial matrix. So mitochondria, like as a chloroplast, is a double membrane cell organelle. It's also we call as semi-autonomous cell organelle. It contains its own genetic material. Inner membrane of the mitochondria can have in many number of the following, that is called as a triste. Inside of the triste, that liquid-like structure we call as a matrix. Matrix. So now you see here in the respiration, aerobic respiration, several steps will be involved. Those are glycolysis, oxidative decarboxylation of pyruvic acid, Krebs cycle, and electron transfer system. So out of this all, glycolysis it takes place in a cytoplasm of a cell. This you can imagine, this is a cell and this is a cytoplasm. Here the glycolysis will take place. First step of the aerobic and anaerobic respiration is the glycolysis. In the cytoplasm, glycolysis takes place, and in the glycolysis molecule, 2 moles of pyruvic acid is formed. These are the 2 moles of pyruvic acid can export from the cytoplasm into the matrix of mitochondria. After the glycolysis, remain all of the steps, oxidative decarboxylation of pyruvic acid, Krebs cycle, and electron transfer system all are take place in a mitochondria. So now glycolysis, cytoplasm they given as a, it can something as a glycolysis, matrix of mitochondria, mitochondrial matrix, Krebs cycle will take place. So grana A2 light reactions, stroma B3 dart reaction, A2 and B3. Grana light reactions A2 and B3 stroma dart reactions, and C1 it should be present. C cytoplasm as a 4 and mitochondrial matrix as a Krebs cycle. So D1. So how to change these options here? A2, B3, A2, B3, C4, D1. Answer A. The next question. Dimorphic chloroplasts are present in. Dimorphic chloroplasts are present in. Here, chloroplast dimorphism, we are here in a concept of the C4 cycle. So, chloroplasts are, that shows 2 types of chloroplasts are we are seen in a C4 plants, that is seen in. See now, what's the meaning of dimorphic chloroplast or chloroplast dimorphism? If you see the structure of the chloroplast, structure of the C4 plants, the C4 plants are, they're having, these are the C4 plants are, they're having a specialized character. The specialized characters are vascular bundles are, they're surrounded by the specialized large cells. Those are the specialized large cells are we call as, they're having tipped cell wall. This type of the cells are we call as bundle sheath cells. And C4 cycle can take place in 2 different types of the cells, that is mesophyll cells. These are the mesophyll cells and these are the bundle sheath cells. So in the mesophyll cells and the bundle sheath cells, Mesophyll cells contain as a chloroplast. These chloroplasts of the mesophyll cells are they contain as a grana. So in these are the chloroplast, grana is present. But bundle seed cells also they are contained as a chloroplast. Which are the bundle seed cells? They have the chloroplast. The chloroplasts are adrenal. Adrenal chloroplast means that grana is absent. So here two types of the chloroplasts are we are seeing into the single leaf means the same leaf that's why these are the leaf belongs into a specialized plant cell. That special plant cell we call as a the special plant it is called as a C4 plant, and the C4 plants are that and perform the two different types of the cells photosynthesis, that is mesophyll cells and the bundle seed cells. So mesophyll cells chloroplast is contained as a grana as usually, but the bundle seed cells are the chloroplast are they won't have a grana. Those are that is called as adrenal chloroplast. So two different types of chloroplasts are we are see. So that's why that C4 plants are they can perform the chloroplast dimorphism. So chloroplast dimorphism is a character of C4 plants, means two types of the chloroplasts are there present. So examples are sugarcane, cotton, mangrove and pea plants. Out of the given four options, we have identified as a C4 plant. So cotton, mangrove and pea plants are they belongs into C3 site, that belongs into C3 plants, and sugarcane only it belongs into the C4 plant that can perform the C4 cycle. So in these are the C4 cycle, bundle seed cells are arranged in the form of a ring-like manner, that is also called as a trans-anatomy. Answer it is A. Two molecules and water molecules and glucose molecules and oxygen molecules is involved, utilized and produced. So now you want to know that clearly. First you want to know the process of photosynthesis. In that process of photosynthesis we are going to represent it in the form of equation. So you know that photosynthesis equation it is 6CO2 plus 12H2O gives rise to in the presence of chlorophyll pigments and light. It gives rise to C6H12O6 6O2 plus 6H2O. This is the molecular equation of photosynthesis. So now question they are asking how many molecules of carbon dioxide, how many molecules of water, how many molecules of glucose, how many molecules of oxygen and the water is produced and utilized. So first you can see as a utilization. Here number of CO2 molecules are 6, number of water molecules are 12 and produced 6CO2 plus C6H12O6 it is transferring as a glucose, means one glucose molecule, and number of oxygen molecules are 6 oxygen molecules are evolved and 6 water molecules also will be released. So now 6 plus 12 18 and here 6 plus 12 6 plus 6 12 plus 13 12 plus 1 13. So here 18 plus 13 means 18 plus 13 will be that as a 131. So total number of the water molecules, oxygen and glucose molecules are utilized and produced, that is number 31. Don't get confused here. We are taken as a 12 water molecules. In that 12 water molecules, 6 water molecules only will be released in now. Why we have to take as a 6 water molecules extra? So here these are the 12 water molecules are required. If the 6 water molecules only you are taking at the time of the balancing of C3 and C4 cycle, number of ATP and NADPH2 molecules produced, it is different. So that's why instead of the 6 water molecules, 12 water molecules are we are going to take in, and here they are mentioning here number of water molecules are utilized. Means first utilized, we have to estimate it, we have calculated, and how many molecules will be drawn to produced. So these are the produced molecules and the utilized molecules. We have to take separately. At the time only will be that as a answer as a 31. If you are not take as a utilized molecules and produced molecules separately, you will be get as a 25 only. But the option 25 is not given. So that's why you have to choose answer as a 31 by this method. And the next question, the visible visible portion of light spectrum useful in photosynthesis is referred as. So for the process of photosynthesis, visible light is required. So that visible portion of the light spectrum useful for the photosynthesis is referred as. That visible light referred as are also called as. They given as option RFLP. That RFLP represent as restriction, fragment, length, polymorphism. Restriction, fragment, length, polymorphism we are seen in DNA fingerprinting. And PAR, photosynthetic active radiation. These are the photosynthetic active radiation, also called as a visible light. That is, we are going to represent as 390 nanometers to 760 nanometers. Otherwise, some of the books they given as 400 nanometers to 700 nanometers. We can consider as any. So means in this wavelength of the light only plants are they can perform the photosynthesis. So that's why we are also called as a photosynthetic active radiation as a part. And then what about the WAM? So WAM represent as vascular or vascular mycorrhiza, and VNTRs, variable number of tandem repeats. So visible light it's represented as a photosynthetic active radiation. In that region of the light intensity only plants are that can perform the photosynthesis. Answer is D.